Right. Well, today we're going to take a look real quick at really the third key question when it comes to discussing cells, which is how do we get more cells? And what this is really going to get us looking at is a lot of the cell cycle and some things that influence what goes on during the cell cycle. So the cell's life cycle, you can really break it down into a number of parts. Honestly, if you talk about cell cycle, we could also say cell division. There's other times that this can be influenced in different ways. For instance, cell cycle, when we talk about it in terms of normal body cells or somatic cells, we're going to refer to this as mitosis. Sex cells, on the other hand, have to undergo a slightly different process because when sex cells get together, you really need to bring together half the genetic component from the mother, half the genetic component from the father, and get that one set so they can't undergo cell division the same way, because if they do, you would actually end up with double the amount of genetic material that you should. And as everyone's seen with like trisomy 21, which is Down syndrome, extra amounts of genetic material don't always work out well for development. So when we really end up looking at the cell cycle can get divided down into a number of different phases. You really have what's called interphase, and then you will see we also have these 4 different phases of mitosis, which is actually divvying up that genetic material that we've copied. Interphase, though, is kind of this normal cell development as well as the beginning stages of the cell cycle, beginning stages of cell division. So most of the time, if you're looking at a cell, it's going to spend a vast majority of its time in interphase, especially those cells that are slower dividing. They're going to spend most of their lifetime in interphase. Faster dividing cells still spend most of their time in interphase, but you're going to see a little bit more time in those other phases as well. And when we break down interphase, we can really break it down into kind of a number of stages. A lot of times they talk about G1, S phase, and G2 phase. There's also something called G0, which is just kind of the normal functioning of that particular cell type. So we're going to take a look at that now. So you can see interphase, when it's doing its normal stuff here, you would kind of call it in G0. What we're going to see during interphase, this G1, S, and G2 phases are really kind of prepping for the start of cell division. And a lot of times when I talk about at least these early stages, if you've ever moved houses or apartments, this is a good example of it. I mean, G1 is kind of some normal function, but also getting ready to divide cells. So when we start looking at G1, we're going to see that you're doing some growth and metabolism. We're also starting to get ready and generate some of the components that are going to be important for cell division. So certain proteins, certain enzymes that are going to be important in these other phases. S phase is the next one. S phase, what we really see going on here is, I always call it the Xerox phase or the copy phase. In S phase, what you're doing is you're making a copy of all the genetic material that is in the cell. So when you divide the cells, each cell is then going to have the instructions necessary to control that cell. In between there, you can see over here, as I can point to right here, you have what is called this G1S checkpoint. And what we end up seeing is this ends up being a really big deal when it gets to kind of normal cell division and preventing cancer. G1S checkpoint is actually a very important part, and a lot of times changes in that can lead to cancer in certain cells. The reason why this is important when you really look at it is that G1 is kind of this prep stuff here. Right after that, you copy all the DNA. There's a good pause in here, and this is a way for the cell, just like you would see at like a shuttle launch, to kind of go, okay, this is propulsion, go. Navigation, go. It's kind of the same thing with the cell here. Is everything ready to go before we copy all this DNA? And the reason why that ends up being extremely important is any change or mutation in that DNA that's not fixed before S phase is no longer going to be just in a single cell. You're going to copy all those mistakes or any changes in that cell, and you're now going to have 2 cells with that copy, and any of their daughter cells will also have that mistake in it. So it's really important that cells kind of take a look real quick between this G1 and S and make sure everything's ready to go before they move on to the next stage. The G2 phase part of interphase, to me, is kind of like that last minute packing. So it's like when you get ready to move apartments and other stuff, like I was saying, you pack up certain things that you're not using right away. It's like if it's the middle of summer in G1, you pack up like the winter coats, the boots, and things like that. But there's certain things you can't pack up right away, which is going to be things like dishes, silverware, things that you really need in order to keep going until you get to that last second stuff that you need to move. You're moving tomorrow. G2 is kind of that. They're doing some little bit of growth, but it's really some enzymes and other stuff that are going to be key in the next steps of that cell division. And those are going to be the 4 stages of mitosis. So mitosis is, now that we've copied all this DNA, we're divvying it up into its 2 separate components. So mitosis, we divide this up into the phases called prophase, metaphase, anaphase, and telophase. And really the last part here, they call this cytokinesis, which is we've already divided all the genetic material, but now we're just separating off and finishing off that division of the actual cytoplasm of those cells. And when I kind of break this down, I always like to use a lot of analogies and other things like that. But to me, when you start talking about cell division and mitosis, I always think about it a lot like what happens when you pick teams at a playground. And Interphase is kind of getting ready to play that whole game. When we start getting into prophase, one of the first things you have to start figuring out if you're going to pick teams on a playground is who is going to be the captains, what people are playing. So to me, that's kind of what happens in early prophase. The centrioles, which we talked about when we talked about components of the cell, those are going to be kind of like the team captains. They start start separating apart to their different poles here, and they're going to kind of control the movement of this genetic material during the rest of mitosis. The other thing that happens during this one is we kind of find out who's playing. That nuclear envelope is going to disintegrate, and those chromosomes, which are a little more diffuse in chromatin most of the time during the cell cycle, now condense or supercondense into visible chromosomes under the microscope. So that happens, and by the end of, as you can see here, by the end of late prophase, you have the centrioles at the 2 poles of the cell, and we have condensed chromosomes. The next thing that happens is, generally, if you're going to pick teams on a playground, you have everybody line up. And that's what happens next here. We see that we actually have, during metaphase, along the equator of the cell, we're going to have these chromosomes that have now condensed line up along the center with those centrioles at each pole of the cell getting ready to separate it out. After that, the next thing that happens is you're going to actually pick teams on the whole playground analogy. And this is what happens here. We now have these centrioles using those little spindle fibers here, grabbing onto these exact copies of these chromosomes and pulling them towards each pole. So it's kind of like picking teams, but it's kind of like your teams were identical twins and each one's getting one set of the twins. So these are then going to be pulled apart to each one here towards that pole. As we get further and further away, we now get to the last stage after anaphase, which is telophase. And to me, during telophase, it's kind of like before the game actually starts. You get everybody together, let's set our plan, let's huddle up. And that's what happens here. We have these now, the nuclear envelope reforming around one set of the DNA in what's going to be each separate cell here. You can see the cells haven't completely divided. And that's where we are during telophase. We haven't completely divided that cytoplasm apart, so we still have this cleavage furrow left. I would joke around and kind of call it the wonder bra stage, because the cytokinesis is kind of lifting and separating that cleavage furrow to separate apart into 2 separate cells. And cytokinesis, again, no more with the genetics here, it's just separating that cytoplasm, and now we have 2 daughter cells, which at this point would be back in interphase. How fast the cell divides, a lot of times is going to depend on what the cell does. So a lot of times, the longer the cell's going to live, generally the slower it divides. Stem cells sometimes are an exception to this rule. They a lot of times will undergo very frequent cell divisions, but more often than not, the shorter the cell lives. So like an epithelial cell, so for example, a skin cell or an intestinal cell, hair, all these things divide rather quickly and generally don't last really long. When you think about your skin cells, they're not alive for a very long time. Other things like nerve cells or muscle cells, which are much longer living, they really don't divide that much. And really where we can see this getting really important to us in kind of a health setting is that cell division and cancer really are kind of one and the same. We talked about homeostasis before. Really, all your different types of cancers are not necessarily different diseases. They're all actually just a disease of loss of homeostasis of the cell cycle. And we see you can get to benign tumors, which, while it's not cancerous, these cells are not acting kind of independent or still doing some normal cell function and other things like that. It is an abnormal overgrowth of cells. Cells are dividing too fast. When we start getting into malignant tumors and cancerous tumors, here we're talking about ones that have now kind of changed enough that what they're doing now is they're actually almost acting as an independent organism. They're trying to outproduce and outdo the normal cells, kind of start invading into these and taking up more kind of thing here. It kind of gets into a survival of the fittest thing here. And what's really interesting is some of the ways that these tumors can happen are directly related to really what's going on in the cell cycle that we went back to. So I talked about already about benign and malignant tumors, but what can cause these a lot of times, they talk about a lot of times in cancer development, oncogenes. What these are is genes that if they get expressed a little bit more, or a gene that was getting regulated somehow is not being regulated as much anymore, sometimes we call these oncogenes. It's kind of like a car getting out of control. It can happen a couple different ways. You could get the accelerator stuck, or you could have the brakes go out. Oncogenes are a lot like the accelerator on that cell cycle getting stuck. It's kind of an overexpression or an overactivation of factors like a growth factor, let's say, that's going to stimulate the cell to divide more often. If you get these oncogenes turned on or overexpressed, I mean, there's a good example in breast cancer of one called HER2/neu is the kind of the name of the genes and stuff that are turned on here, but what it is, it's an overexpression of something called epithelial growth factor receptors. It causes these breast cancer cells, and it's not a real common type, but it causes these breast cancer cells to overproduce and lead to tumors because you're overexpressing this oncogene, this receptor for this epithelial growth factor. Another good example of genes that can cause this are tumor suppressor genes. So when I was talking about that G1S checkpoint, a lot of those genes that are regulating, that are really forming the proteins that are part of this G1S checkpoint, they are what we refer to as tumor suppressor genes. These are ones that normally cause a cell cycle to slow down, to pause, take a little bit of time. If these tumor suppressor genes are lost, what happens then is it no longer takes that pause. It goes barreling through that, and divisions happen a lot quicker because we've now kind of cut the brake lines on that cell cycle. And what's really kind of interesting on this one is this actually is what, when you start talking about chemotherapy and other stuff like that, a lot of times it actually goes after cells that have lost these tumor suppressor genes. So when you really start talking about chemotherapy, a lot of times these are mutagens. These are things that lead to mutations in cells. And the whole idea here is these cancer cells, they don't really pause. They don't fix things. They just keep going through as fast as they can. And if you go in there and actually mutate a gene by using chemotherapy that is really important for the survival of that cell, it's not going to take the time to fix it, and therefore it won't be able to keep surviving. So the whole idea with chemotherapy on a lot of these, or radiation, is to kind of cause these cells that are no longer pausing and fixing things to get damaged enough that they die off, where our normal body cells are going to try and fix that. Again, though, I mean, it really is, really, when we start talking about chemotherapy or radiation, it's a lot of collateral damage for what we're trying to do, but as of right now, it's kind of still one of the best ways we can go after these cancer cells. Again, actual targeted therapies directly to the cancer cells only would be ideal, and they are working on stuff that can start doing that, but as of right now, kind of especially with most cancers, kind of these chemotherapies and radiation therapies are still most of your first line kind of treatments that we're going to see for cancer. The other thing that happens with cells is they go through the cell cycle a lot of times. They're going to differentiate. So these are cells that get much more specialized. As we start out really early on, we started as a single cell and we're not specialized at all. The device of two cells, either of those two cells can make an entire human. As we go further and further along development, we start getting this differentiation and we start getting cells that are going to be kind of determined to become a particular, like a skin cell versus a liver cell. And what's really neat on this is it's just a different set of genes turned on or off that lead to each of these ones here. The other thing that's kind of interesting is generally the more differentiated cells are, the less likely they are to form cancers. So it's another one of those that's kind of interesting. So what we'll pick up with next time is we'll start talking a little bit about how cells communicate with one another and what's going to be important there.